Hi, my name is Michael Fox. I'm pleased to give a brief talk today on causal mapping of depression symptoms using big data. So the question we're very interested in my lab and have been interested in for some time is the question of how do we localize neuropsychiatric symptoms? How do we figure out what location in the brain or what circuit in the brain is responsible for a specific neuropsychiatric symptom? And for us, this is not just an academic question. We want to identify a therapeutic target. If we can localize a neuropsychiatric symptom, then we can intervene to help that symptom. We can implant deep brain stimulation electrodes in the location of the brain to make that symptom better, or hold a TMS coil over a location in the brain that can make that symptom better. And the most intuitive approach for localizing neuropsychiatric symptoms is neuroimaging of symptomatic patients. You take patients that have a particular symptom, such as depression, you put them in a scanner, and you see what functional imaging abnormalities you can identify that differentiate a patient with depression from a patient without depression. Or maybe you identify areas where brain activity correlates with depression severity. Now, this is a very effective approach, and this approach has dominated the field of functional neuroimaging for a long time. But there's one problem with this. And that's, in the end, no matter how big you increase your sample size, no matter how large you increase your data set, in the end, this approach is only looking at symptom correlates, not causes. Now, correlates of depression can be fine if what you're looking for is an imaging biomarker that can predict who's likely to get depression versus not. But if you want a therapeutic target, if you want an area in the brain where you can intervene to make depression better, then this issue of causality becomes critical. And there's actually a very finite number of causal sources of information for mapping out human brain function. By far, the oldest, and I would still argue most valuable source of information is focal brain lesions. When a patient gets a lesion to a particular brain region and then develops a new neurological or psychiatric symptom, there is a causal link between the location of that brain damage and the new symptom. Now, newer forms of causal information for mapping human brain function include brain stimulation, things like deep brain stimulation or transcranial magnetic stimulation. So let's start by focusing on brain lesions. Now, one of the best ways for mapping out human brain function in a causal way is to find a lot of patients that all have focal brain damage causing similar symptoms, in this case, patient 1, 2, and 3. You map out the location of their brain damage onto a brain atlas, and you just look at where these lesion locations intersect. And that spot in the brain where they all intersect gives you a very strong causal link between that location of shared brain damage and the shared symptom. However, when you actually try to map human brain function using lesions, more often than not, this is what happens. You take lesions in different locations that all cause the same symptom, you map them all out, and you're left scratching your head trying to figure out where the responsible brain region is. Well, enter in a very valuable resource that is, I believe, going to transform neurological and psychiatric research and care, which is the human brain connectome. So for the last couple decades, the field of neuroimaging has been collaborating to build a wiring diagram of the human brain, either based on where water likes to diffuse, that can give you a rough map of where the anatomical connections in the human brain are, or based on spontaneous fluctuations in blood oxygenation that reflect spontaneous brain activity that allow you to map out functional networks in the human brain. And all of these initiatives give us a wiring diagram that tell us how different brain regions are related to one another. So now let's return to our problem of mapping out human brain function. And now, rather than ask which brain regions do these lesions intersect, we can ask whether these regions intersect a common brain network. And for this first time, we can map lesions causing the same symptom but occurring in different locations onto a common neuroanatomical substrate. And we've done this for a variety of different symptoms. It turns out that this has been a very effective approach for mapping out a lot of different neurological and psychiatric diseases. But today we're going to focus on just one. We're going to focus on depression. And so Jaya Padmanabhan actually turned to big data. Depression was not an easy syndrome to map, and she ended up putting together 5 independent data sets from 3 different continents, a total of almost 500 lesions that either caused depression or were not associated with depression. Jaya then mapped out the location of all this brain damage and just asked, is there any spot in the brain that, when damaged, is significantly linked to depression? And the answer was no. She then turned each one of these lesion locations into a map of all the brain regions that are connected to that lesion location using the human connectome. She then asked not where are the differences in the lesion locations themselves, but what are the differences in the brain circuits that these lesions are damaging? She then got a very robust result, which was that lesions associated with depression were connected to the left dorsolateral prefrontal cortex, while lesions not associated with depression were not. And what that means is that you can look at connectivity with this one location in the dorsolateral prefrontal cortex, and that defines a distributed brain circuit that, when lesioned, will lead to depression. And so what you see here is a map where lesions to the yellow areas will be associated with depression, while lesions to the green areas will not. Now, in this particular network, this was exciting because there had actually already been 3 trials of brain stimulation for post-stroke depression. And the location that they stimulated in these trials are shown here with these 3 colored dots in the left dorsolateral prefrontal cortex. Now, this was exciting to us because what it suggests is that when we use brain lesions that cause a symptom to map out a brain circuit, that same circuit might be a valuable therapeutic target for making that symptom better. So let's turn from brain lesions to brain stimulation, another form of causal information. And just like with lesions, where the brain damage is linked to a new symptom, in the case of brain stimulation, the stimulation site can be linked to symptom relief. And so let's start with transcranial magnetic stimulation, which has been used extensively in depression, but it doesn't work for everybody. And so the patients that were stimulated at these green locations got better. The patients stimulated at the red locations didn't. And we had a long-standing hypothesis that maybe the sites that work better are the ones that are connected to limbic regions, like the subgenual cingulate. And that turned out to be true. And so we actually could predict how well people were going to do just based on where their stimulation site was. And in fact, we were very excited to see this replicate shortly after our study, where the same relationships were observed in an independent cohort in Australia. Again, the more connected the stimulation site was to the limbic system, the better people did. And so you could argue that you can use this to identify an optimal antidepressant target where you map out the circuit and then use that circuit to optimize your brain stimulation treatment. But we noticed something important, which is that this circuit target wasn't equally good for all symptoms of depression. As you got closer to this juicy blue spot in the dorsolateral prefrontal cortex, sadness got better, pessimism got better, loss of interest got better, but other things like sleep, irritability, appetite, they actually didn't improve as much. They went the opposite direction. And so Sean Siddiqui in my lab started mapping out what are these sets of connections you have to hit to make these different symptoms better. And there was one set of connections for loss of interest, a different set of connections for past failure, a totally different set of connections for changes in sleep. And as he mapped out all these different symptoms, he noticed something important, which is that when you drop the threshold, the same set of connections seem to be important. It was the same set of connections for sadness, anhedonia, concentration, but a very different set of connections for interest in sex, sleep, and indecisiveness. And he ended up identifying 2 circuit targets for 2 totally distinct clusters of depression symptoms, a dysphoric cluster and an anxiosomatic cluster. And he could use this data, and this is big data. There were a couple hundred patients that were stimulated in these data sets. And he could map out their response and actually create a map on the surface of the brain of where you need to stimulate to help dysphoric symptoms and where you need to stimulate to help anxiosomatic symptoms. And he actually went on to validate this in an independent brain stimulation data set. And again, when you look at the scatter of the brain stimulation sites in these different data sets, and that's shown by these black dots here, you could actually predict which symptoms were going to get better in which patient just based on where their stimulation site was on Sean's targeting atlas. In fact, when Sean turned to the literature and looked at how well these different symptoms have done in different clinical trials, he could even predict which types of symptoms were responding to different TMS clinical trials. Now I'm going to leave you with one thought, which is the real big data approach to precision psychiatry, or really identifying that brain circuit target for depression, is to combine brain lesions and brain stimulation all together in one analysis. And so Sean Siddiqui's next project was to take 14 different independent data sets of lesions, TMS sites, DBS sites, put them all together. So 465 lesions, 151 TMS sites, 101 DBS sites, map out the connections with every one of these either lesion locations or stimulation sites, and identify a brain circuit target based on those connections. And they identified one circuit target for depression based on lesions, one circuit target for depression based on TMS, and one circuit target for depression based on DBS. And you can see across these three different independent approaches to causal mapping of brain function, they all identified almost exactly the same depression circuit target to try and get depression better. So I'm going to leave you with three conclusions. One is that lesions, DBS sites, and TMS sites allow for causal links between human neuroanatomy and symptoms. A map of human brain connectivity, this wiring diagram, is required to make sense of this causal data. And by leveraging the human connectome, we can identify the causal substrate of depression symptoms and hopefully improve our therapeutic targets. I want to acknowledge brilliant people in my lab that contributed to this work, but also a huge number of collaborators that have generously shared their data sets with us so that we can do these types of analyses. And please reach out directly to me with any questions that might come up as part of this talk. And for those of us that are at Brigham that might be watching this talk, we've launched a new Center for Brain Circuit Therapeutics, and we're happy for collaborators and happy to get as many people involved as possible. So thank you for your attention.